This is our neuron. Some key parts of the neuron include the axon, which sends the signal to other neurons, myelin sheaths that protect the axon and allow for saltatory conduction, the cell body that helps send the signal, and finally, dendrites that receive signals from other neurons. Now let's take a closer look into an axon.
Neurons by default maintain a membrane potential of negative 70 millivolts. This is kept constant by sodium and potassium pumps, which pumps sodium out and potassium in, maintaining a concentration gradient. Generally, there is a high concentration of potassium inside the cell and sodium outside of the cell, so to maintain the gradient, energy from hydrolysis of ATP is used to provide energy for the pumps. The ratio of potassium in to sodium out is 2 to 3 because the positive sodium acts on both a concentration gradient and electrochemical gradient, as the negative inner cell attracts the positive sodium. For this reason, more sodium is ejected because it enters at a faster rate. It is important that this potential is maintained because a change in potential could open voltage-gated channels, which could lead to signals being sent when they were not meant to be sent.
When there is an influx in sodium, the cell depolarizes and an action potential is created. In this model, the influx of sodium occurs as a result of a ligand opening a ligand-gated channel, which allows for more sodium to enter the cell. If the influx of sodium is able to change the membrane potential to be negative 55 millivolts, voltage-gated sodium channels open up, allowing for more sodium to enter. As the positive charge is spread down the axon, subsequent voltage gates would open, allowing for even more sodium, creating somewhat of a domino effect that carries the action potential down the axon line. When the membrane potential exceeds a certain threshold in a region, the voltage-gated potassium channels open up, allowing for potassium to exit the cell, thus making the cell more negative. The voltage-gated sodium channels are also closed, preventing more sodium from entering. The potassium exiting repolarizes the cell until the membrane potential hits negative 80 millivolts, where the cell is now hyperpolarized. This period of hyperpolarization is known as the refractory period. The refractory period is the time following an action potential when a second action potential is unable to be initiated. As the action potential moves along the axon, the refractory period prevents the production of another action potential behind it. This prevents action potentials from traveling backwards towards the presynaptic cell body and forces the action potential to travel in a one-way direction.
Nodes of Ranvier located between myelin sheaths or Schwann cells carry out action potential using voltage-gated sodium channels. These nodes transfer the inward current produced during the rising phase of action potential to the next node, where it depolarizes and initiates another action potential. This mechanism is known as saltatory conduction, where the action potential essentially jumps along the axon from node to node. The action potential then arrives and depolarizes the plasma membrane of the synaptic terminal. This causes calcium ion gates to open and triggers an inflow of calcium ions, which cause the synaptic vesicles containing neurotransmitters to fuse with the presynaptic membrane. The neurotransmitters are then released from the synapse terminal of the presynaptic cell and diffuse across the synaptic cleft, where it binds to a ligand-gated ion channel in an excitatory response. The neurotransmitters cause depolarization of the postsynaptic membrane, which moves the membrane potential closer to the threshold.